Okay, now that we've seen the origin of our bile components, and we've briefly examined the function of the bile as lipid emulsification agents in the intestine, we have to get the bile to the intestine. So let's take a look at bile secretion. Now recall the architecture of the hepatocyte. The architecture of the hepatocyte is very important to its function. We're going to take a look at two hepatocytes, two cells that are next to each other. Now the hepatocyte is a polarized epithelial cell. Polarized. So what does that mean? Polarized in the same manner that we saw the acinar cells in glandular secretions. So that means that the apical surface here is going to be facing the portal circulation. The area above this, and we'll look at this in more detail, the area at the apical surface of the hepatocyte is actually comprised in an area region that's called the space of Dis. The space of Dis is a separation between the endothelial cells that line the hepatic circulation and the apical surface of the hepatocyte. So your portal supply, your portal vein material, which is coming from the gut and digestive system, mesenteries, is coming through here, and the endothelium here is fenestrated. And fenestrated means that the top of the epithelial cell, the hepatocyte, actually has free access to the circulatory supply here. Now the next structure we have to look at is the basal surface. And the basal surface is where secretion occurs that is going to end up into the blood. Now what is this structure I've drawn right here? This is called a bile canalicular space. This is where the bile is going to be secreted because this is going to be continuous with the hepatic duct that leads to the gallbladder. So this is where the material is coming into the hepatocyte from the portal circulation. Material from the hepatocyte will be secreted into the blood through the basal surface, but bile components will end up in the canalicular ducts. Now how do we get that in there? Well, because bile is so dangerous, and dangerous I mean that it's antipathic structure, we need to get it out of the liver cell as soon as it's secreted because it could solubilize the liver membranes. And so how we're going to do that is to couple the bile to a transporter, and this transporter is going to be an ATPase transporter. So this is going to be primary active transport. We are going to hydrolyze ATP and utilize this stored energy to drive the bile from the internal cytosolic compartment of the hepatocyte into the canalicular space. Now, once we have that in there, we need to get this down to the gallbladder for storage. And the way to look at this is a very simple drawing. If this is the bile source meaning the liver, this would be the common hepatic duct. This leads into the cystic duct, which of course leads into the storage of bile, which is the gallbladder. Now, under normal conditions, the liver is secreting the bile, going into the canalicular space, All of these ducts will coalesce into the common hepatic duct. This will come down the duct and then enter the gallbladder through the cystic duct. Now, why doesn't it go in this direction? This would be the duodenum. Now, we said that the bile has to get into the duodenum in order to digest help digest the lipids. However, what we need to do first is build up a storage of bile so that we can secrete a nice healthy load into the duodenum when a high-fat meal comes upon us. So what we're going to do here is we are going to direct the bile into the cystic duct to be stored in the gallbladder. But what prevents us from exiting down this way? Well, what we have here is a small sphincter. This is called the sphincter of Oddi. Under normal resting conditions, this is going to be closed. At rest, which also means in the fasting state. So in between meals, the sphincter of Oddi will be closed, preventing any of the bile secretions coming out of the hepatic duct from exiting into the duodenum. Therefore, it only has one place to go, and it can move up the cystic duct and be stored in the gallbladder. Now, what also is happening here is these ductal cells here coming out of the liver into the common hepatic duct are also secreting bicarbonate. We saw this in the salivary gland, and the purpose of the bicarbonate here has the same properties that we want to have, for example, in the pancreatic secretions. We want to decrease increasing the bicarb into the duct along with the bile secretions. What we are going to want to do here is to increase the pH of the bile solution, the bile secretion. And what this is going to effectively do is to increase solubility. Increase solubility of the bile components. And it increases the solubility of the bile components because recall the pKa's of our functional groups on the bile salts. The higher the pH, the further away we get from the pKa, which means the greater the ionization. Increasing ionization increases solubility. Now, once we get the bile into the gallbladder, we are going to continually make bile components, especially the bile salts in the liver, so we are going to continually secrete from the liver and store in the bile in the gallbladder. So in order to do that, we need to concentrate the bile that's in the gallbladder. So in order to do that, let's take a look at the gallbladder membrane. So if we draw a cell that is lining, an epithelial cell that is lining the inside of the of the gallbladder, what we're going to see is that the top, if we draw this in orientation so that this is the lumen of the gallbladder, we are going to have on the basolateral surface our typical sodium, potassium, ATPase, And this sodium-potassium ATPase is going to drive sodium out of the cell. And I think you know what will happen when we drive sodium out of the cell and into the interstitial fluid. Water will follow by osmotic pressure. So the concentration of the bile is not so much increasing the concentration of the bile components, although we are doing that, but we are also decreasing the concentration of water. Now this can cause for a little bit of a problem, because as we increase the concentration of bile salts, remember they have those hydrophobic domains to them. Those hydrophobic domains can start to associate to try to escape the aqueous environment, and when that happens, we can form a stone. So what prevents that? Well, as we're taking away water, what we're going to want to do is increase the concentration of bicarb moving into the lumen of the gallbladder. And this is conveniently coupled bicarb chloride antiporter. And so now we have a path for a chloride channel to exit into the interstitial space and neutralize the sodium that's coming through the sodium ATPase transporter. Now, what happens when we move into a fed state? So now we've eaten that hamburger with a nice load of fat in there. Now recall originally what is happening in the digestive tract. We have our sphincter of Oddi, which is under fasting conditions is closed. Our gallbladder is storing bile that is being fed from the common hepatic duct. And now we are going to rely on the hormone called CCK, cholecystokinin. So cholecystokinin is secreted during digestion. We saw this in the intestinal secretions, pancreatic secretions, that the cells that are responding to fats and amino acids are the enteroendocrine cells, and they are secreting CCK in response to food, to lipids and amino acids. These open type enteroendocrine cells regulate the secretion of CCK. CCK is known hormone we saw in pancreatic secretions to be able to stimulate the secretion of enzymes, digestive enzymes, in the pancreas. It also has a dual role here in that it can bind to the smooth muscle cells that create the sphincter of Oddi and cause them to relax. So thus the sphincter of Oddi is going to open. The other mechanism that CCK can do is it can bind to the smooth muscle cells around the gallbladder itself and cause this to contract simultaneously as the sphincter of Oddi is relaxing. So in this way, the bile that is stored can now exit through the common bile duct and enter the duodenum. Now recall there's another duct that comes in here, and this is the pancreatic duct. And I bring this up here because this particular area in here, where the common bile duct and the pancreatic duct come together, is called the ampulla of Vater. And this is simply like a vestibule where these two ducts come together. And the reason I put this in there is because of the beautifully coordinated activity of a single hormone, CCK. So recall CCK is being secreted from cells recognizing the lipids and amino acids, and they have a multifunctional purpose of first causing enzyme secretions into the duodenum. Simultaneously, it's allowing the relaxation of the sphincter of Oddi in order for all of these secretions to come through, and it's causing contraction of the gallbladder to get the bile into the duodenum to emulsify the fats so that the pancreatic enzymes that are coming through and mixing have the greatest level of efficiency.